Well, so can everyone online hear me just fine? Just write in the chat if you can't, because I have the chat pulled up on my screen. Thanks everybody for joining me today. I'm excited to talk to you about the delinquent late-night pollinators. Here's a little roadmap for my talk. I'm going to do a little introductions. Since next week is Bat Week, I want to share some bat facts with y'all. Just get you excited about it. Maybe it might be some new things you don't know, or if you've gone to any of my other talks, I apologize, there'll be repeats of that. I'm going to talk a bit about my research and how it applies to agave and bat mutualism and other research that's been done on that. Talk about some of like the active other research outside of plants that I'm doing and the preliminary findings I have, and I'll have some time for questions at the end. If you have any burning questions, feel free to ask me during the talk. Okay, fine. Okay, so starting with introductions, I'm Mallory again. She, her pronouns. I'm in the 4th year of my PhD, almost done with it. And I'm at Colorado State University. And as I said before, on my intro, I'm now a research scientist at University of Arizona, so I'll be transitioning over there, starting in May, hopefully finishing my PhD at the same time by like next fall. Yeah. My PhD research is assessing the drivers of long-nosed bat movement at the northern extent of their range. Oh, feel free to come in. I'm part of the Stoner Lab. We need more chairs. Okay, so I'm part of the Stoner Lab at Colorado State University, but my advisor over here, Catherine Stoner, recently started as the director of the School of Natural Resources at University of Arizona. So that's why I'll be moving over there. So Stoner Lab is now based out of there. You might recognize some of these folks. This is Scarlett Seller. She's a former master student of Catherine's when she was at New Mexico State University, and she's currently finishing her PhD there. This is Rachel Berg. She has been working for the Forest Service at the Gila Ranger District, Gila Wilderness Ranger District. And she'll be moving over to Bat Conservation International to be the new agave coordinator, so agave restoration coordinator. And this is Teresa Laverty. She was a postdoc in the lab when I started, and then she's now a professor. She's the new mammalogy professor at New Mexico State University. So we've all helped contribute to the research that I'm going to present on today. And then these are current lab members. Kennedy is joining us in the audience over here. She's a recent graduate from Colorado State University, has been working out here with me for the past 6 months. Is hopefully gonna get a Forest Service job for next year. Sorry, got a few calls. Catherine again, and Daniel Milton's my other technician. He's been with me for 2 years now as an intern in my lab and is helping build some really cool radio equipment that we're gonna use to track bats with. And these are my past field crews. If some of you have seen me before, you may have met some of them.
Thank you.
That's not, I don't think that's the camera that's looking at me. Okay, so now we're going to go over some fun bat facts. This can be very interactive. Feel free to ask me questions in the middle of this if you want more clarification on the things I'm going over. So, what are bats? Oh, bats are mammals. I didn't need to push the button. Bats are mammals just like us. They're placental mammals. So, what is, so you can come in, you're fine.
Thank you.
Yeah.
Sorry.
No, you're okay. They're out of seats. There's
That's okay. I can step on
steps.
steps.
I don't know if there's seats nearby in classroom that we can feel.
Steps.
Okay. So bats are placental mammals just like us. Let's go back to that one. That means they give birth to live young. They feed their young milk. They can regulate their temperature. So bats have been on Earth for more than 50 million years. They're the second largest order of mammals next to rodents. So 22% of described mammal species are bats. They can be found on 6 continents, excluding the poles. And they're the only mammal that can fly. And some bats hibernate, and some migrate. Bats are of the order Chiroptera. Chiro is Greek for hand, as in like chiropractic. And ptera is Greek for wing, kind of like pterodactyl. So you put it together, it's winged hands. So I like this diagram. It's a human arm, hand, and then a bat. So a bat's wing is attached from their fingers to their ankles. The little blue thing is their thumb, second finger, third finger, fourth finger, fifth finger. And then the wing goes down all the way to the ankle. So if you kind of think of it, it's elongated fingers. So they're wing hands. So you see little cartoons, little bat hands on top of their wings. That's not accurate. That's not how it works. It's just their thumb. But they can grab stuff with their thumb. They're always like grabbing things. It's annoying because they're like getting stuck and fighting back. There's two suborders of bats. There's Microchiroptera and Megachiroptera, so microbats and megabats. There are no megabats in the US. We only have microbats. And microbats are generally insectivorous bats, but they can feed on, they have a variety of diets as well, where all megabats eat fruit. Nice, you found a chair. So it looks like we have extra chairs here. If people are sitting on the steps and want to have a seat, you're more than welcome to move. Megabats generally don't use echolocation, or actually megabats don't use echolocation, but microbats do. So echolocation is the high pitch ultrasonic sounds that are produced by the mouth. The sound waves come out of their mouth and then they hit the objects and bounce back. And generally they pick up the bounce back with their ears, but there's some bats that pick it up with their nose or their face or other parts of their body. I think it's mostly just nose and face though, and ears. Oh, and then cool thing about echolocation, at the Southwestern Research Station, just over in Portal, Arizona, so pretty close to here, there's a researcher who did work on a moth that is like adapted to jam bats' echolocation. So they have echolocation jamming. So some really cool local research. I'm not sure if that moth comes all the way up here into the Gila, but if you're down in the Chiricahuas or Palencios, that moth is probably confusing a lot of bats. So bats live in a variety of habitats. These bats are underneath a palm leaf. So it's like they're like the tent bats, Honduras tent bats, I think. And so you're kind of like hanging upside down. So it's like a peak if you're like lifting the leaf up and seeing. Some bats live in like rotten logs, in cavities created there. Some roost in like abandoned mines. Bats I study roost in cave sites. These are the long-nosed bats that I work on. Some roost on tree branches and others in cacti or yucca. And roosting is just where a bat sleeps during the day. Bats also have a variety of diets. There's insectivorous bats, bats that eat nectar and pollen, fruit bats. Some bats are carnivorous, so they'll eat small mammals and birds. Some eat other bats. Some bats eat frogs. Patagonia, one bat eats frogs. Some eat fish, and there's also bats that eat blood, so vampire bats. And they have all these different adaptations because of physical, morphological, or behavioral characteristics that allow them to live in different environments. The cool thing about these bats that eat like fish and frogs, I want to share because I don't go as in depth with it as I normally do in my other talks, but they use echolocation to sense the ripples in the water to know where to fish or to find the frogs. And so they'll echolocate towards the water, it bounces back these ripples, and they know. And then the ones that eat frogs can actually tell different types of frogs, like poisonous frogs, apart from non-poisonous ones, based off of their mating calls. So bats are pretty cool. So you know, in case you haven't picked up on that. So why are bats important? They provide really important ecosystem services, like outside of their intrinsic value, which that's good enough for some people, but not for everyone. Bats eat a lot of insects that destroy crops. They save agriculture thousands, possibly millions of dollars every year in pest control, and it limits insecticides that we would have to use, or pesticides that we would otherwise have to use on our crops. they disperse seeds and they can help forests grow. So those fruit bats are eating those fruits and then they're pooping all over the place. They're just flying kilometers and spreading seeds all over forests. We have the pollinators, so that will also, they pollinate many plants. So pollinator bats will eat the nectar out of it while they're foraging on the nectar. Their face is all up in that flower and completely covered with pollen. They fly several kilometers to another flower and do the same thing and are transporting that pollen from one flower to another, which is spreading genetic diversity. So far, way further than any bird or bee that would probably be doing the same, would spread that pollen. But many of these bats are in danger of extinction. So the ongoing destruction of natural habitats such as cave sites that they may live in, hunting and persecution for sport and meat. So also out of fear, during the COVID pandemic, a lot of bats died because they were falsely blamed for spreading the disease or the virus when we found that they probably weren't the cause. It would be very unlikely for them to happen. But before that, there's a lot of fear and bias against bats. So folks were killing bats by the thousands, governments included. The growing use of wind turbine energy is killing off bats. And this is something that I'm going to try to focus a bit more with my research because there are some interest in putting up some wind energy or wind turbines down in the Blue Heel of New Mexico and the Animas Range particularly. And the issue with wind turbines is, for both bats and birds, is that they tend to kill them in large numbers. And it's not just because of direct contact. So like, think about how large those blades are when you see it coming through on a train or a truck. And they're bigger than this room. And then when you're on the highway and you're watching them turn, you can kind of see the blade moving. It's kind of moving, like the tip of the blade looks like that speed. But if you're thinking about the length of that thing and how fast it must actually be moving to look like this from far away, I don't know how fast it is, but it's really fast. And it's fast enough that it changes the pressure of the air surrounding the blade. So there's this drastic air pressure change from right in front of it to right at the blade or surrounding the blade. And these wind turbines are set up in areas where there's really good wind, which also overlaps historic migration routes. So birds and bats would fly where there's really good wind, so they don't have to work as hard to get where they're going. So it's already overlapping historic migration routes. And then for bats, they emit a high frequency sound that draws them towards it. So this drastic pressure change, if the bat were to, or bird were to approach it, just entering that change of air pressure causes their lungs to explode and they drop to the ground. We call that barotrauma. So the bat or bird never actually has to come into contact with the blade. So it's set up in their major migratory pathways and it emits an ultrasonic sound. So bats are actually drawn towards it when they're migrating through those areas. So it's unfortunate, it's killing off a lot of bats. I'm not against wind energy as it like an alternative fuel, but I do think there needs to be changes with it. There's been a lot of research to try to do lights or ultrasonic sounds or other things to try to deter bats from flying towards it. Unfortunately, none of them are working at a scale that would actually make a difference. So I'm at a loss for how I feel about it right now. But I think it's important to not put up wind energy, since we know that this is a problem. We shouldn't be putting up wind energies in major migratory pathways of birds or bats and birds that are already endangered or threatened, because it would cause major population decline while they're migrating through. And maybe one day we could like pause the wind energy when the bats and birds are migrating through, because we have ways to forecast that now. So we could put a pause on it and have them running not during peak migration periods. So kind of like how they have the lights out warnings for bats and birds that Cornell University pushes out. So just a thought, things I'd like to work on more in the future. Proliferation of harmful myths cause fear for bats and then people kill bats instead of just letting them out of their house. And climate change is degrading habitat. So bats are running out of habitat. I think it's about 1/3 of all bat species are currently at conservation or considered at risk for population decline, are currently threatened or endangered. Okay, so now after all that depressing stuff, I'll tell you, I thought usually I do it interactive, but I want to get more into the science on this talk, so I just have some quick bat facts that I'd like to share with you. So this is a common bat myth. I crossed it out because I didn't want anyone to get confused because I had bat facts written on top. But a lot of people think that all bats have rabies, or that a large number of them do. But transmission of disease when encountering a wild animal is a common concern. However, it's now thought that less than 1% of bats are rabid. Vampire bats do have rabies at a disproportional number because they can live with rabies. And since they feed on blood and they have a communal feeding strategy, which is really cute, they like, if one goes out and feeds and gets a good meal and comes back, and their buddy, not even related, didn't get a good meal, just like their buddy from their roost that they hang out with, they will actually regurgitate blood and share their meal with their friend, kind of like a mama to a baby bird. So, oh, that was great. There's only 3 types of vampire bats. I don't think I have a vampire bat slide. I forgot. I meant to leave it in. But there's 3 types of vampire bats in the country. None of them are in the US. They're mostly in South America. 2 feed primarily on avian blood. So they don't feed from mammals, 2 of the species. And only 1 feeds from mammals, mostly ungulates and cows and livestock. They have been known to feed on blood from ranchers. They don't suck blood. They just make like a little slit, usually on their back or the ankle of the ungulate. And they have an anticoagulant in their saliva. So when they make that slit with their incisors, they modified incisors, the blood doesn't coagulate, so it continues to bleed, and they kind of just like lick it up like a cat drinking milk. So it's not that bad, but rabies does transfer from blood and saliva, so you get it. So from saliva to blood is how rabies transfers. So it's like the best way for them to transmit that virus. The world's smallest mammal is a bat. The bumblebee bat is less than 3 inches long. It's like a hand, so it's pretty cute. And the world's largest bat is the giant golden-crowned flying fox. So you want to try to take a guess at how long its wingspan is?
18 inches.
A bit bigger.
8 feet.
A little bit smaller than 8 feet.
5.
Yeah, I think I heard it. It's a 6-foot wingspan. So I'm 5'8", so I have like a 5'8" wingspan, let's say. So it's a little bit longer than mine, but they're not as tall as us. Like bats have really tiny legs. So it's like this, but they're still like, if you look up pictures, it's crazy. They look like vampires. Like it's probably what vampire cartoons are based off of.
Where do they live?
Oh, I looked it up. It's somewhere in Asia. Do you remember, Kennedy? We were like looking at the pictures. They're somewhere in Asia. I can't really remember
Not
where.
Australia?
There are flying
Not-
foxes in Madagascar.
Yeah, there are in Madagascar. Teresa Laverty, that professor, actually studies them in Madagascar,
Yeah, there are some in Australia.
in Australia, and in Africa.
In the Philippines.
In the Philippines, thank you. Yeah. So pretty big bats in the Philippines. There's a lot of really great places for ecotourism, for visiting forests and stuff where flying foxes will converge. So if you're curious about that, you could ask me, or it's pretty easy to find online because people over there really enjoy it. The fastest bat in the world is the Mexican free-tailed bat. We have those here. Those are the ones that you'll find on the bridges and other places, in those very large colonies, like at Carlsbad. I think it's the Brazilian free-tailed bat there, but still, they're very fast as well. So they can fly up to 100 miles per hour. And bats hold the record for the oldest small mammal ever recorded. There's this tiny bat from Siberia. It's a France myotis, and he holds the world record for living over 41 years in the wild. So they caught him 41 years ago. He was already an adult when they caught him the first time. Recaptured him again 41 years later. This was like 10 years ago. They haven't recaptured him since. Probably not likely that they will, because 41 is really old to begin with for a tiny little bat. But it's pretty amazing, because usually smaller mammals live shorter lives, but bats have this really long lifespan. Bats are important pollinators. They pollinate over 528 plants. They're one of the main pollinators for most of those. This is including 300 species of fruit. So some of those fruit that we all enjoy are like mangoes, bananas, cacao, peaches, cloves, and agave. So in New Mexico, there's approximately 30 different species of bats in the state. And I'm going to highlight just a few before I get into the meat of my research. So I get a lot of calls or have a lot of questions about this big blonde bat that's roosting in their doorway and pooping everywhere. And there's just like poop and insects on the ground right in front of your door. And they have these like long ears. And if you have ever experienced that, it's a blonde bat, long ears, lots of poop and insect parts on the ground below them. It's probably a pallet bat. Pallet bats are really cool. They go for ground dwelling insects and arthropods that other bats ignore. So arthropods are small animals without a backbone and have an exoskeleton like crickets, grasshoppers, beetles. Pallet bats will also eat spiders, cicadas, scorpions, and the desert centipede. So they actually hunt on the ground, they'll get on their little elbows and crawl and attack scorpions, and they seem to be immune to their venom. And the other centipedes are supposed to be like poisonous to eat, to ingest, but it doesn't seem to bother them. So if you have these little blonde bats, the big ears in your doorway, if they're pooping a lot, I mean, at least they're keeping the scorpions out of your house. I think it's a plus. I have some living in my attic, and we haven't had a scorpion in our house all year, so it's great. Or maybe one this year. Usually, they seem to like adobe structures or attics. Like, I don't really know how. You can have a bat box, but out here, bat boxes don't really work because there's so much good natural habitat, which is a great thing. I have to think on that. There's also Brazilian free-tailed bats, like I said earlier at Carlsbad. Approximately 400,000 bats emerge every night. And just think about how many insects they must feed on and how much that must help agriculture in that area. And then there's 3 nectar bats in New Mexico. So 3 nectar bats persist in the US, or in continental US, and all 3 occur in the state, which is really special because I don't think you would say that for any of the other states. So these are the bats that I study. There's 2 long-nosed bats and 1 Mexican long-tongued bat. The long-nosed bats are in the Leptonicterus genus. There's the lesser long-nosed bat right there. It was recently delisted back in 2018. So Catherine Stoner's lab, the lab I'm in, is in charge of the post-delisting monitoring. So delisted means they were taken off. They were on the endangered species list. Both these bats were put on the endangered species list at the same time. And this one was taken off the endangered species list due to recovery. So it was like a good thing. It was the first bat to be taken off the endangered species list. And that happened in 2018. And when we do take a species off the endangered species list, we do what we call post-delisting monitoring. So usually that goes for about 5 years. But with this bat, because of how their population is, we're doing potentially a 16-year post-delisting monitoring. Still in draft form, and I'll have 16 years confirmed once they give us the final form. But so we're at year 5 now, but we will probably be continuing this research for another 11 years. And then Leptonicterus nivalis, or the Mexican long-nosed bat, is still currently listed as an endangered species. And it occurs, last year I was able to confirm that it still is here using eDNA, or environmental DNA, at the Big Hatchet Mountain Race where I do most of my work. Oh, I had it all on a slide, so sorry. But since everything I just said, so it was removed from the endangered list in 2018, first bat species removed. And then Mexican long-nosed bat, or Leptonicterus nivalis, is currently listed as endangered. And there's the Mexican long-tongued bat, Pollinators mexicana. It was put up, there was a candidate for listing as an endangered species back in 1991. However, by 1994, they deemed it data deficient. This bat occurs out here. We caught one 2 years ago, and I think I detected one on trail camera, so I need to look back and confirm that. But this is something that I'll be focusing my postdoctoral research on. I want to collect data, see if their populations are at risk or not. So some of you might have these guys coming to visit your hummingbird feeders at night. And if you want to know how to tell them apart, I have a few tips. So they have different tail membranes. So for long-nosed bats, I'm gonna call them leptos. And for the long-tongued bats, the choreo, the one that's not listed that's data deficient, I'm gonna call them choreos. Just because the other one is too confusing, like long nose, long tongue, it gets confusing. So leptos are the long-nosed, that's the 2 species. Choreos, long tongue, data deficient. And the leptos will be on the right and the choreos will be on the left for all these pictures. So you keep it straight. So they have different tail membranes. We like to say that the leptos have pants. So you'll see there's this gap right here between their legs, where the choreos have skirts. They have a tail membrane connecting their 2 feet. They have different looking snouts. So the leptos have this like short, cute dog looking snout. They're adorable. Choreos kind of look like a horse. Kenny thinks they're ugly. They have a longer snout. Their body size is different. You can't really tell this too much from sight, but just, you know, leptos are about 20 to 35 grams for the ones that we catch out here. And then choreos are 10 to 25 grams. They're a bit smaller, but their wingspan looks the same. It's just their body is like tinier. These are a bit more muscly. They have different life histories. So Leptonicterus is known to roost deep into caves and mines in large colonies of 1,000+ individuals. Their choreos tend to roost in like the entrance of caves or like crevices and canyons and rocks. And they're generally in smaller groups of like 1 to 13. So that's why they're harder to study and they're data deficient, because they're just everywhere, spread out. They're not concentrated at these roost sites where we can go and study large groups of the population. Leptos, so the Gila National Forest is the most northern distribution of their range that we've recorded so far. Jennifer Fry just published a paper this past year citing that. She found one up in the northern region of the Gila. They occur in Arizona, New Mexico, and Texas. Only one of the leptos are in Arizona, but both species occur in Texas. Yerba Buena only like accidentally there though, like in El Paso. And then choreos can be found in several other states. So they're found in Nevada, New Mexico, Texas, California, and Arizona. So they're a bit more spread out and they don't seem to be like such like stuck on certain roost sites like the leptos are. And here's some just really cute pictures of leptos that I've caught before I get into like the researchy stuff. Okay. So on a half hour and doing good on time. So talking a bit about agave and bat mutualism and kind of relate it to my research a bit. So as I was saying before, our lab's research focuses on the 3 nectar bats. But for this talk, I'm going to be talking mostly about the lesser long-nosed bat. So the leptos. They're important migratory pollinators and seed dispersers. So they promote healthy soils, diverse vegetation communities, and create sustainable economic benefits through, in Mexico, they're called the tequila bat. They're the primary pollinator of agave. So they're the reason that we have tequila to begin with. So they support tequila industries. Long-nosed bats are migratory bats characterized by resident and migratory subpopulations. The resident subpopulation remains year-round in southern and central Mexico to mate and give birth, while the migratory subpopulation, consisting of mostly pregnant females, travels approximately 1,200 kilometers north each spring to give birth in maturing roosts in northern Mexico and southwestern United States, following a hypothesized nectar corridor of flowering cumular cacti and agave species. So, resident population hangs around Chiapas mostly, but they intermix genetically with this other population that's considered the migratory populations. They're not super distinct, but there's one that sticks behind and it tends to be the same individuals, but they do breed with the individuals that migrate. So they're still the same groups kind of. So they migrate, pregnant females migrate up to northern Mexico and Arizona to give birth to their young in early spring. And then for some reason, come over to New Mexico before heading south. And some of that reason is that the arrival of bats to this region here is thought to be coincide with the regional flowering of agave species, which is the only nectar plant food source known to be used by long-nosed bats in this area. And we think that they come for add this extra leg onto their trip before going south, because agave has these two really important amino acids, one that supports lactation and one that supports muscle development. So lactation for the lactating moms, so they can feed their young, and then muscle development for the young, so they could build strong muscles to be able to make the trip back home. But that doesn't really explain why they're here right now, and the agave have been done blooming for months. But ignoring that part, reason I'm here, agave, I'm gonna go a bit into agave since this is the Gila Native Plant Society. I spent way too long making these extra slides. They're not even that nice looking. Did this just for y'all. So there's the agave are part of the Asparagiaceae family. So the asparagus family. That's kind of why they look like asparagus when the stalk is first emerging. Is there anyone here that doesn't know what agave is, or is this your first time seeing it? It grows all over town, right? So we've all seen it. If you didn't know it, that's agave. It's also called the century plant. Is another thing you might have heard it referred to. It takes 8 to 30 years for them to fully mature and flower. It really depends on the water, the amount of water it receives, I think. You don't really know why they flower yet, like what sets off the flowering, if drought or if extra rains or what. But maybe I could disentangle some of that information from all the data I've collected. They're monocarpic. I said that wrong, someone feel free to correct me. That means they flower only once in their lifetime, and after flowering, they produce fruit and then they die. So take 8 to 30 years to flower. So it's really tragic when a flowering stalk breaks. It's like, oh, that was your whole life. And now you're broken. Agaves, when they're immature before they're flowering, they have a basal rosette. Their flowering stalk is considered paniculate out here because they have the arms. There are some that are spiked, where it's just a spike, kind of like sodal. Yeah, kind of like a sodal. But we don't have those in this region as much. We do have some. Is it Lechikia? I know Shaddai agave, but those are more in crevices, and they're at such low amounts that I don't really include them in my analyses because I haven't been able to find any. Also, so I haven't been able to track them. So the lateral branching that they have distinct umbels at the end of each branch. So those are groups, bunches of flowers. There's about 150 green tubular flowers that point upwards and release nectar shortly after nightfall. And the umbels begin to bloom on the bottom of the branches, and they work their way up. So flowers at the bottom start opening first to the top. Sure, y'all have seen that. Here's some flowers I collected from my clipboard and took a picture of from fall and agave. So that's the bud. You can actually cook the buds up and eat it. They do that in some parts of Mexico. I don't think they use palmeri or perii agave. There's a different one, but apparently it's edible. This is, it has 2 different phases. So agaves are protanderous. I said that wrong too. Apologize. So that means the male reproductive organs come to maturity before the female. So this is a flower in the staminate stage. It's a flower that's pistillate. And this is one after it's been successfully pollinated and is turning into fruit. So although this looks like the flower might no longer be producing nectar, it is still very much producing nectar. So it produces nectar for about 10 days is what I've noticed in this region. Other books say it's like a week, but it seems to be a bit more than a week. We do weekly surveys on all the agaves and the flowers are still fresh. There's some overlap week to week. So day 1 through 4, the stamens and anthers develop and release pollen. And about day 5 through 10, the stigma emerges and it becomes receptive, so it can't pollinate itself. So it depends on a biotic factor to pollinate it. They're not self-pollinating plants. In case you wanted some things, so those are anthers. This is where the ovary is, the tepals, stamens, style stigma. Sorry, I forgot I said anther already. I was looking for that. I'm not sure. There are two dominant agave species that persist in this area. It's the agave palmeri and agave perii. So perii is the one that you have more in town. There are surprisingly some groupings of palmeri that grow out in the hills. Rachel Burke told me. I haven't actually seen them yet. So they're a little bit of a different life history. Agave palmeri, as you can see, is huge. It can be huge. These girls are my height. This is our first field season out in the Boot Heel, New Mexico, over in the Little Hatchets. And the basal rosette could be anywhere from 1 meter to 2.5 meters wide. They're typically about 1.5 meters, 2 meters on average. The stalk can be 4 to 8.5 meters tall. This is from our data that we've collected on hundreds of agave on the field. But on average, the stalk is about like 7 meters tall, I think. I think so. Yeah. The lateral branching, both agaves have about 11 to 30 arms from our data, with a 20 arm average. 1 to 3 umbels. Okay, this is a big one. On the palmeri, only 1 to 3 umbels open at a time. They'll like open per week. So it takes about 8 weeks to fully open. So as an umbel wilts, another one opens. The agave perii, all the umbels open within a week. I don't know if you've seen that in town, but like once it starts opening, it just kind of all opens. And it's really beautiful. But then it only blooms for about 2 weeks. I don't know if you've seen something different in town. If you have, I'd love to hear about it. This is how the agave perii goes in the Boot Heel of New Mexico. About 2 weeks and then they're wilted. The agave perii that grow in the Boot Heel have a basal rosette of like 0.5 meter to 1.5 meters, but it's usually around 0.5 meter or 1 meter wide. The stalk grows 2 to 4 meters tall, or a bit shorter. The umbels are bunched a bit together. So it's all a bit like kind of more condensed. People usually like it, they think it's prettier. They tend to plant it in their yards more often, but it's still a great resource for the bats. So palmeri is the dominant in lower elevations in southwestern New Mexico. So blooms last up to 8 weeks. So the bloom usually starts in early June and goes through late September. The last agave just finished blooming a few weeks ago. Where perii is the dominant agave here in higher elevations. Blooming can last up to 2 weeks, and then it blooms early June to early July. So on the first palmeri are opening, and there's very little nectar on the landscape. Perii serves this spot where it produces a lot of nectar on the landscape for these bats coming in to take advantage of. However, perii in southern, where I work in the Boot Heel, New Mexico, isn't studied nearly as much as, or the high elevation agave is studied not as much as the lower elevation one, because it's really hard to get to this. Kenny and I just went and did our final hike to collect our trail cameras that we had deployed on all the agave on Big Hatchet Mountain, and it took us like 12 hours. So like get all the way up there, get all of our trail cameras and get back. We're kind of fast hikers. It's not an easy hike. So a lot of researchers haven't done this in the past. So this was kind of a missing piece of data from the puzzle. So I apologize if I said this right, but due to the absence of flowering cacti in New Mexico, the agave plant serves as the only nectar plant food source for nectivorous bats in New Mexico, or is suspected to be. And the reason we only are looking at like cacti and agave is because both those plants, or mostly agave, has the chirolithic pollination syndrome. Have anyone heard of pollination syndrome when thinking about hummingbirds and the flowers? So with hummingbirds, you'll have those trumpet-like flowers where the flower is elongated, and they have this high sugar content that goes with the hummingbird's metabolism or metabolic rate. So for nectar bats, so the agave species exhibits morphological features that are congruent with chirolithic pollination syndrome. So chirolithic meaning bat loving. So chiro, you remember, is like the hand wing. So the floral shape and size, so the flowers are upward facing, they have light petals. So the moonlight will make them glow at night. It has this large inflorescence event where it throws up this huge flowering stalk so the bats can find it, because nectar bats find their food through sight. They look for the agave stalk or the flower on the cacti glowing in the moonlight. Then they use smell to figure out which ones are fresh. So they could smell the sugar content coming out. And then they just use echolocation through their nose. So they're the philostomid bats or the leaf-nosed bats. Use this little thing there. They echolocate through there and it bounces back. So they know where to put their little face into the flower to feed from it. So the floral shape and size, so upward facing yellow petals. Nocturnal nectar production starts producing nectar around like 8 p.m. Most of these agave from studies that have been done in the past. The nectar quality contains the proteins and amino acids that I mentioned before that are important to the bat's diet. I think I didn't mention the proteins, but pollen, the nectar has proteins in it, so that's important versus just the sugar water and hummingbird feeder, which is just sugar. The nectar quality, and then the high sugar concentration, it ranges from 18 to 26%. And that overlaps the bat's range of 5 to 29%. So nectar bat characteristics that go with this chirolithic part is that they have this elongated rostrums, their nose and tongue, to be able to get the nectar out from the bottom of these flowers. They have an accelerated metabolism, and that's how they're able to digest those sugars without going into like, I forget what it is, but the thing that diabetics get when they have too much sugar, if they're that type of diabetic. So Rachel Burke, who I previously mentioned earlier, she, for her master's thesis, mapped out the seasonal distribution models. She tested the spatial temporal nature of the nectar corridor hypothesis. So these bats follow this nectar corridor of cumular cacti and flowering agaves as they make their migration from southern Mexico up to the southern US. So this map, so she tested the nectar corridor hypothesis with independent data of plant species richness. So ones that these bats are known to feed on that are part of their diet. And she determined that species richness of food plants is a major driver for migration and distribution for all 3 nectivorous bats found in the US. So these maps are seasonal distribution models for Leptonectarus yerbabuena, so the one that was recently delisted, the lesser long-nosed bat. The map shows suitability of lesser long-nosed bats based off of nectar food availability. So winter, spring, summer, fall. However, as shown here, the distribution of these bats is most concerned during periods of migration and distribution in the US in the spring and fall. So they're a bit unsure. Had those on this one. Distribution is most concerned during spring and fall, and that suggests that factors other than feed plants might influence migration patterns, but it also confirms that they do follow it during other periods. In this recent study, I spent way too much time reading today to try to put this poorly constructed slide together. It just came out like a couple weeks ago, and it's on the historical, temporal, and geographic interactions of the 2 nectar bats, so the 2 leptos and agave. And they constructed 3 co-presence matrices between agave and the 2 long-nosed bats to explore possible dynamics of their biotic interactions. So you're doing stuff from a really long time ago, from the last, what do they call it? The LGM, the last glacial maximum, and the LGI, it probably wasn't G. It was the thing that happened before the last glacial maximum. So in between the ice ages that we've had, so last ice age when it was really cold, and the time before that, when in the period that we're kind of mimicking right now, the inner something. And they inferred the historical presence of the 2 agave interaction corridors. So this one here is the lesser long-nosed bat, the one that was recently delisted, and this is the Mexican long-nosed bat, the one that's still considered endangered. They were able to find historical presence of these 2 interaction corridors with stability from the last glacial maximum to the present. using these historical, temporal, and geographic interactions that they built into this matrice. And they also were able to find agave leptonictus interaction refuges in these areas in the Tetrakan and the Metzitalan Valley. Sorry, I said that wrong. And some of these interactions using paleontological, geological, and molecular data suggest that these interactions may have remained active for the last 2.5 million years. So that's, I didn't know those bats were even around then, so I don't really understand that, but they probably were. But the LGM that they found with these interaction corridors, that was from 20,000 years ago was the LGM, or last glacial maximum occurred. So those are also extremely old. There's other studies that have been out that I don't think I cover here, but I want to stay on here because this map is nice, where they overlaid historic nectar bat ranges, historical ranges, with paniculates, so the agave with the long arms, historical distribution. And they found that where the bats ranges were historically, not like even now, some of them went extinct or have moved, but that's where paniculate agave still is. And where there aren't historical bat ranges is where the spigot or the spiked agaves are. So there's theories out there that the bats, their faces got longer, tongues, their like rostrum nose got longer, their tongues got longer as the agaves grew larger. And if you're thinking these bats find the agaves with sight, they're going to be looking for these really large flowers. So the larger the flower, the more likely the bat is to take that pollen and bring it to another agave. So bats started selecting for larger agaves, where the agaves also, the bats would do better if it had longer faces to get into these flowers. So it's kind of like the agaves were selecting for bats with longer faces. So they had this coevolution and this mutualistic relationship where as the bats' faces got longer, the agaves got larger, and then agaves started putting all their energy into this one large flowering event because they were more likely to get, well, they didn't start putting in, but like the bats selected for the ones that did that. And there's this Mexican researcher that likes to say that the bats domesticated the agaves so we can tequila. Because without them, agaves would all look kind of like yucca. So that brings me to the whole agave and nectar bat mutualism. So because of this chirolithic pollination syndrome that I went over earlier, so nectar bats' anatomical and physiological characteristics, this geographic co-occurrence, that historical range that the last paper just talked about, where it dates back, where they can find these interactions from super long ago. And the temporary correspondence between agave flowering and the nectar bat migration timing, something that Rachel Burke studied with the species distribution models that she made, overlapping and showing the bats' movement through those corridors during those time. All that points towards agave and nectar bat mutualism, which indicates a coevolution and a mutualistic relationship with nectar bats that pollinate it. So agave and nectar bats having both those things. However, this means that the loss of the food plants along the long-nosed bat migration corridor could jeopardize both agave and bat populations. So they both highly depend on each other. Without one, the other one will fail. So although the tequila industry might be able to get by on clones for a while, eventually disease would wipe out all the agave that industry, which is a very large one in Mexico, depends on. Okay, so that was my thing on agave and bat mutualism. Now I'm gonna get a little bit more into like my research and why I'm here. So leptonectar species need two things. Well, two main things for conservation. We need a lot of other things, but these are the things that we can focus on. So the presence of suitable roost sites. So they really are attached to these roost sites where they have 1,000+ individuals that come, and then they migrate to them every year. And the availability of forage resources, such as agave in this region. So climate change. So but agave is threatened by a lot of things. Sorry to start talking. So climate change threatens agave. Tequila production, although I keep talking about it, actually threatens agave. Livestock grazing threatens it. And wind farms threaten the bats themselves. You can quick touch on a few of these. So agave, agricultural, time is it? So the agriculture of agave is an issue because when agave matures, it starts to shoot, or for the production of tequila, you actually harvest agave right as it's starting to shoot up its stalk. So it'll start to shoot up its spigot or stalk, and they cut it off immediately because at that point, all the sugars are leaving the leaves and they go into the heart of the agave. And they cut off all the leaves on the agave and they're stuck with like this big ball or the inside the heart of the agave. And that's what they use to roast, turn into mash, and make tequila out of, or they smoke it for mezcal. That means that they're converting a lot of acreage or hectares of land into a monoculture of crops that they aren't letting go to flower. So this was an issue. And then they were planting agave to replant their fields from the clones that they shot off right before they started to like try to shoot up their stalk and cut it. So they'll harvest those clones or pups is what we call them. And they would replant the fields those next time. So now these are all becoming genetically similar. So it's not only like a monoculture of the same species, they're also a genetic monoculture, which makes them very vulnerable to disease. So if they all have the same immune system, they all, well, immune is a plant, but they all have the same genetics, they're all susceptible to the same disease. So one disease could come out and has come out and wiped out entire crops. And it takes 10, 15, 20 years for these crops to fully mature. So it's 10, 15 years of work that these farmers have put in, money, effort, for their crop to fail. So it's not good for the farmers either. So not good for the farmers, not good for the bats. So researchers in Mexico are actually working with tequila companies, tequila producers, and they came up, they did some studies and found that if you leave 5% per hectare of crops, of agave crop to go to flower, it would produce enough nectar to support the bat migration, the population that we currently are estimating to come through. And for farmers that participate in that, letting 5% per hectare of crop go to flower, the researchers will come in, they collect the seeds from those crops into flower and turn to seed because the bats all foraged at them. And they will grow those or redistribute the seeds. So either they go to a nursery and give them plants, or they redistribute those seeds for the farmers to grow. And now they have these more genetically diverse plants and a more resistant crop to disease. So it benefits both. And if you're interested in supporting, like if you like to drink tequila or mezcal, you're interested in supporting this, you just have to look up bat-friendly agave. And there'll be a list online of all the bat-friendly tequilas and mezcals, and they have a little sticker on it. It has a bat. So it's nice. It's a little bit more, but it's usually they're nicer agaves too, so it's worth it, or they're nicer tequilas. Oh, and then livestock. If you haven't heard any of the agave plantings with BCI, you may have heard why livestock is an issue. Livestock is not direct consumption of the agave. And it's not just the livestock's fault, it's that we're always in droughts out here. So we're in the northern region of the Chihuahuan Desert. We've had a lot of droughts recently. Last year was different. It rained a lot, but it almost rained too much to the point where stuff rotted because everything was constantly flooded. And the boot heel can like withstand, our botanist who works for the BLM once told me that a boot heel can withstand two things, withstand drought and it can withstand overgrazing, but it can't do both at the same time. So because both are happening at the same time and grazing hasn't been relooked at, the AUMs or annual units that can graze per month, we're starting to see erosion happening. So like if you look at the creosote, they'll start to be pistling out. So you can see that there's soil erosion happening. The grasses are dying out. You can see there's not a lot of grasses in this photo that I took in the boot heel. Obviously this is like a sacrifice plot because it's where the cows are and there's a salt lick over there. So this is like an extreme example. But agaves really depend on having those mother plants. So small bushes, grasses, other things to provide it shade to hide it from the sun during its first few years of development until it gets large enough to have its own like water stores to be able to support itself through periods of drought. So without the shade, the agaves are drying out and dying. So we're losing generations of agave during periods of drought. And they're not really direct consumption. Sometimes the cows will eat the stalks, but it's not really anything compared to the recruitment or the young agaves not being able to make it and become established. Talked about wind energy already. climate change is another thing. This paper came out in 2019. So this study showed that there's a 75% reduction in suitable habitat for agave within the distribution of Leptonicteris nivalis. So Leptonicteris nivalis, the endangered bat, is in blue. Light green is distribution of agave. Dark green is where the two overlap. So you can see a clear nectar corridor going up here to northern Mexico, southwestern US, we're here to Texas. We have a roost, Nivalis has a roost in Big Bend National Park. Using the most conservative RCP value, which is the relative carbon something, someone else remembers, but it's just like carbon emissions or carbon that we would produce. And this is the most conservative value that people generally, climate scientists generally tend to model. By 2070, there was a 75% reduction in suitable habitat. So you'd see dark green is only down here. There's no more nectar corridor going up to southwestern US. Nectar corridor is very sparse going up to Texas. So given changing weather and climate may affect the timing of the phenology also, like so the timing of flowering of these plants and its reproduction. It's possible that the presence of long-nosed bats in the US, the timing of agave food availability might not line up anymore, which threatens the future viability of both of these species. So there's already signs that there might be a potential phenological mismatch between the presence of bats and agave nectar availability, as some of you might see like tonight, if you look at a hummingbird feeder and the bats are here, there's no agave flowering. So agave blooms in New Mexico typically peak in July or early August, while bats tend to start arriving earlier in June and depart by late October. So this suggests that fewer nectar resources may be available to bats in the spring or early fall, and indicates a change in resources between seasons. This is everything I just said. Sorry. I had given a talk for kids recently, so I had all my slides up here as well as talking through them. I forgot that I did that. So these are some diet studies. So we want to look into this, like, what are they eating? Are they eating insects? So the diet, like, are they eating insects? Are they just eating sugar water? And this one is looking at arthropod consumption, and it seems to be in relatively low amounts. So, but this most of these studies occurred in Mexico, South America. So Arizona being one of them. That was in the 70s. It was published in 79, but data's from the 70s. So that's kind of outdated with everything that's going on right now. So diet of long-nosed bats, especially in terms of arthropod consumption at the most northern extent of their range, is not yet fully understood. So that's where I come in. So my research objectives are to look at this, like, why are they here? We have this mutualistic relationship with agave, but they're here and the agave aren't here. What are they eating? Are they eating bugs? We don't really know. So I'm going to look into that. So I want to analyze the seasonal and annual variation in diet at multiple sites in relation to the density and availability of nectar. I want to evaluate the influence of artificial nectar availability has on the variation in diet. I want to evaluate movement patterns and site occupancy in relation to nectar availability in diet. So of it's looking at agave in relation to how their diet changes and where they are on the landscape. Like, are they near these residential areas taking advantage of artificial nectar feeders, or are they at Big Hatchet Mountain Roost, where there's, like, I know of like 3 hummingbird feeders that are within a 50-kilometer radius of that roost site that people maintain. So we came up with some hypotheses. So I have the diet plasticity hypothesis that lesser long-nosed bats will supplement their diet with alternative food sources during periods of low nectar availability. So as nectar availability decreases, insect consumption would increase. And as agave food accessibility decreases, so plant nectar food availability, bat populations will increase in areas where known artificial nectar food sources are and decrease in areas with none. So there's 2 parts to my study that I'll talk about today. I'm gonna definitely talk about the diet analysis. If I have time, we'll go into movement, but I'm getting close to an hour right now, and I do want to leave some time for questions. But I can usually get this talk in 15 at a conference, so I think I got this. So, okay. So we examine the diets of individual long-nosed bats using fecal samples and pollen samples collected from captured individuals, and fecal samples collected weekly from plastic ground sheets that are deployed in the cave that we work at. A subset of fecal samples are sent off to a lab, but I'll get into that in a second. I think I'm jumping ahead myself right now. So here is a beautiful map I made that I'm very proud of. So I put in all my presentations now, but these are all the sites where we study movement also. These are get covered later, but these are pit tag sites. So we tag bats, just in case I don't have time to get to it, with chips, kind of like you would microchip your cat or dog. We just insert these tags subcutaneously between their shoulders. There's no battery. It's just kind of like an antenna, basically, that has a unique ID. So they get excited by antenna and it will record that number. So we have antennas set up at known caves and mines where these bats will roost. And we're working with 12 different researchers. So we've been tracking them all throughout this range. And the ones highlighted in yellow are where I study. I'm doing these more micro movements in the southwestern US. Get it closer. Sorry, those are my study sites. The stars mark foraging sites. The circles are roost sites where I work. Oh yeah, in Silver City, these are all the houses. A lot of you are in the audience, so thank you so much for being part of my study. The main roost site I work at is on Big Hatchet Mountain. So Kenny and I were on Monday or Tuesday, all the way up there. That was tiring. But our roost is down here somewhere, so we only have to walk halfway up the mountain once a week. Equally as tiring. It's not a good trail. So to test our diet plasticity hypothesis, we examine the diets of individual long-nosed bats at this roost site based off of using fecal DNA while monitoring agave nectar availability within a 50-kilometer radius of this roost site at Big Hatchet Mountain in southwestern New Mexico. So we do a 50-kilometer radius because the bats are known to forage about like 100 kilometers in one night for agave round trip. We're returning to the roost, so 50 kilometers out, that's 100 kilometers round trip. Newer papers that came out since I started my study are now saying 70, but that seems not viable. So I'm sticking with 50 for my radius. Big Hatchet Mountain Roost is the largest known roost site in the state of New Mexico and the only roost site known to have all three of these nectar bat species anywhere. So it's really special for that reason. It's believed to be a late summer transition roost where long-nosed bats, mostly female and their young, stay prior to their migration south. And the fecal samples that we collect, so we'll miss net, or we'll set up a net right over here, camp out there overnight, catch bats. We collect fecal samples from bats in hand and we do pollen swabs to their face. And here's that same slide again where I actually start talking. So we'll pollen swab the bats, get a little pollen off them. And we deploy a sheet weekly and collect samples off of that. So we can get incidental samples as well, in case we can't catch bats because we're not always very successful at this site. Subset of those fecal samples that we collected are sent to Pisces Molecular Lab in Boulder for genetic analysis to confirm both the bat host species and identify plant and arthropod species in their diet. So what is DNA metabarcoding? So we take the poop, we put it in a tube, send it to them, they do some fancy stuff, the field sampling, they extract the DNA from the poop. We have 3 primers that we tested against, that we perform PCR on. So we do mitochondrial DNA, we use a COI primer. For chloroplast DNA, we use a tRNA liner or primer. And for mitochondrial DNA, again, we do a COI primer. So mitochondrial for bat, for arthropod, and then chloroplast DNA for plant species. All that goes through a machine. Once we get the data, they sequence it. We use bioinformatics. It's like a program that'll read the DNA sequences and there's this whole inventory of them, catalog. They use the NCBI blast. And they're able to say, oh, this is a 99% match this DNA for this species, so it's probably that species. And I get it back with the number of reads. So I receive a spreadsheet that identifies the species or identifies the species or higher taxonomic level in case you can't figure out what the species is. And then the number of reads per sample, and recounts the number of matching sequences recorded during sequencing, but reads can change based off of PCR efficiency. So it's not really a great number to go off of for like analyzing your data, but I still can tell you some information. Like if there's a lot of reads, it was there. There's just a little bit of reads, you're like, maybe it was there, maybe it wasn't, maybe it's just like a bug crawled over it and like pooped, and now I'm getting the bug's poop in the diet. So yeah. Oh, push the wrong button. So we sampled Bathos plants fused to arthropod, and in 2019 we had 44 uncontaminated samples. 2020 we had 49. 2021 we had 43. I'm supposed to get 2022 results on Friday, so I couldn't include in this talk. I'm sorry, I don't have it yet. I'm kind of bummed about that because of how much rain we had. It would have been interesting. Oh, and I'm not going to include 2019 in a bunch of the data because I don't have phenology data for that yet. I need to figure that out later. But to assess nectar availability, we established and monitored 5 agave phenology transects in 2020 and 2021. Transects were visited weekly to track individual plant phase. So in 2020, we had 5 agave phenology transects, followed 131 flowering individuals, visited them weekly. And we said like percent open, percent fresh flowers, and like when it stopped flowering, so we can kind of get an idea for nectar availability across the landscape. 2021, 5 transects again, 117 flowering individuals. Last year we had like about 300 individuals. This year we had like 400. It was insane. Maybe 300, I don't know. It was a lot. For all of these maps, 2020 data, which was a drought year, if you remember, will be in dark green. I said maps, I meant graphs. 2021 will be light green. The green on this is pollen from agave. And these are reads, so that thing I said that can be variable based off of PCR efficiency. This is a point where like, there's a lot of agave here. And I think the reads tell us that there's a lot of agave here and there's not a lot of other stuff going on. So this is where reads can give us some information, but it's not something I could say concrete. It's not like actual proportional for what was present in their fecal sample. But green is pollen from agave, reads from agave, and gray is other. And I went through every single plant, and every single other, I had like actually 4 categories that I classified the plants into. One was agave, one was dispersed through wind dispersal. One was abiotic, but by daytime pollinators, or biotic daytime pollinators, so like birds and bees. And then I forgot what the other one was, but oh, and then one was bats. So agave's bats, but also I wanted a separate category for other things other than agave. So it's like cacti or other nighttime flowering plants that would like produce nectar at night. So ones that the bats would target intentionally. I didn't have any of those in 2020 or 2021. Had some of those at a different site though. So I know I can pick them up. I just didn't because it wasn't here. And I'm curious how in 2022 because there's so many flowering things. I don't know. So every sample but one in 2020 and 2021 contained agave. The one sample that didn't was right over here. And that's because the agave was basically done blooming. I think that was just out of luck. So to revisit, here is the green. I took out that slide, but the green is from our agave phenology surveys. This is percent of plants out of the total number of plants we surveyed that were open on that day, to make it as simple as possible for this talk. So, and this is only for agave palmeri. Perryi actually in 21, yeah, which is the light green, peaked over here. So that's not being counted. So there's actually like kind of a spike over here that should have been bimodal. 2020 is the dark, 2021 is light. We would predict that they'd be consuming a lot of insects here because there's low agave availability, except for 2021, because we don't have that peak here. There's bimodal, so then that's different. Not a lot of insects during the agave peaks. Can't really see it, but here's the 2021 peak right here. It's a little bit delayed in the 2020. A lot of insects at the tail end because there's no more agave on the landscape, so they'd be supplementing their diet. And then if I look at percent samples that had insects in the diet, black is 2020, gray is 2021, darker color 2020, lighter color 2021. 2020. Oh, 2020 had a peak in insect consumption at the beginning when there's no agave. 2021 had a decrease, but if we put in the peri-eye, elevation agave, that made sense. Decrease both at the peak. And all the samples during low nectar availability contained insects. So in total, 79% of my samples contained insects overall. So this is super high, and you might think it's super high because it was from DNA metabar coding, so they were able to detect more, where these were from microscopic analysis. But if we take in Scarlett Seller's work, that master's student that I introduced in the very beginning, she did microscopic work on DNA fecal samples from New Mexico and found 49% of the samples using microscopy alone had insects. So it kind of indicates that there's a high consumption of arthropods in New Mexico. There's also the argument that arthropods could be consumed due to incidental consumption, so they're feeding on the nectar from the flower. There's a bug in it, they eat it. Now they poop out a bug, and I detected. So I want to break up all the bugs into different categories. Bugs that are associated with flower in any stage of life are green. Bugs that could be found on bats, like mites or other things that you'd find in a cave, gray. So those might accidentally be consumed incidentally while grooming. So not like targeted, but they ate the mite. Blue, all stages of life, aquatic. So they're always found in water, can't incidentally be consumed from a cave or from a flower. Yellow is others, so that's like ground dwelling arthropods. And light gray is unknown. It's like too high of a taxonomic classification for me to really figure it out. And it was really cool. In 2020, during the drought year, and only during the drought year, did I find aquatic insects in their diet. So I found aquatic Caloptera, shore flies, fish flies, freshwater pelagic copiopods, freshwater ostracods, and water mites. And I had some trail camera footage obtained by the BLM that showed them feeding from water troughs during that drought year, where we normally don't in this region. And it kind of suggests that they are using water sources and potentially targeting water sources with protein, like insects in it. Where, because they are captured feeding from waters in non-drought years, but I'm not getting insects in their diet during those times. We also found hide beetles, green lacewings, brown lacewings, net-winged insects, grasshoppers, common desert centipedes, and spiders, which all these aren't really associated with flowers. Some of them feed on nectar, some of them feed on the bugs that feed on the nectar, but they wouldn't really be, they're really big, they wouldn't accidentally be consumed. So these arthropods are either ground dwelling or flying, and they wouldn't be consumed while instantly foraging on nectar. So here's my summary for the diet. Southwest New Mexico agave is the primary nectar plant food source found in the long-nosed bat diet, so it's still the primary. Arthropods make up a significant part of the diet for long-nosed bats in this region, so I think they're supplementing their diet with arthropods later in the season. Consumption of arthropods occurs at high rates throughout occupancy, and detection of the aquatic ground-dwelling net-winged arthropods suggests intentional consumption, and the observation of the bats gives us more evidence for that. And I should be out of time. There's effects of drought that I'm happy to talk about. That's going to be a talk I'm giving in a couple months. I just want to, I'm doing movement and migration using thermal cameras to get the population size at Big Hatchet. So this is a thermal camera, count the bats. So how Kenny started working for me, she would count bats on a computer for hours a day. We get the weekly population. We collect fecal samples. Like I said, we have the pit tag logger, which I touched on earlier. SM4 use acoustic monitoring, thermal camera, and weather data. So we're gonna try to model the populations and see why they're there with the agave nectar. And then I net at residential sites. I hope next year to deploy this pit tag reader. This is my prototype that Daniel made, picture of it. And then play this while these are bats visiting this prototype. I was hoping to deploy it this year at residential sites to get more of those micro movements, but unfortunately the bats are skeptical of the rain and they won't put their heads through it. So we're gonna work on a fair ride antenna now. That'll hopefully be able to read the bats, and we're gonna try to deploy that next year. So if this is stuff that you're interested in getting involved with to try to figure out these micro movements, so I wanna track it as the agave stops producing in the boot heel. I wanna see, are the bats then moving from the boot heel right at Silver City? Because they kind of, as soon as they disappear from Big Hatchet, I start getting phone calls and emails from folks out in these areas saying the bats have arrived, they're back, and in high numbers, and they're making a mess and they're draining the feeder. So like if you ever come, or if you put a full hummingbird feeder up at night, hoping to like feed the birds in the morning, you wake up in the morning and it's empty, and there's a big sugar mess on the ground. It's probably these bats bumping into the hummingbird feeder, spilling the sugar water everywhere and drinking it. And I want to be able to track individuals and see where an individual moves through. Partially because these wind energy developments, I need to figure out where those migration corridors are of these bats to see where I could better advise managers on where to put these developments to avoid these important migration paths. And also we want to know, like, Bat Conservation International are doing all these like really cool agave plantings, but where should we put them? Like, should we keep planting agave perii out here? The bats aren't really here when the agave perii are blooming. Is that actually going to help them? Or is it a wasted effort? Should we be planting maybe palmeri that blooms later in the year? Are there other agave species that could be planted that will bloom even later than palmeri that we could have out here that would actually supplement these bats' diet? Because when the bats stick around and they're just feeding from the hummingbird feeder, I like to call them town deer. You know, skinny and scrawny, underweight, like really long forearms, really low body weights. Their forearm to body weight ratio is off, which tells you poor body condition, covered in ticks and mites. They don't look great. They shouldn't still be here. So to leave this off, go through all that. It doesn't matter. But how to get involved. So if you want to get involved, sorry, this is really small text. You can monitor agave with the National Phenology Network. I can give you more information on that. But if you look up the National Phenology Network, it's there. Do agave plantings with Bat Conservation International. Cool thing is, Rachel Burke and I work very closely together, and she's the new agave coordinator. And she did her whole master's thesis on this. So she's probably the best person for the job. So she's aware bats being here this time of year isn't good. Agave planting, when you're planting agave that don't bloom during the right time of the year, might not be that beneficial for the cause that you're trying to work towards. So she's excited to work on that and excited to work with anyone interested in it. And then if you want to host a residential site during the 2024 season, contact me. I was not very reliable this year with getting back to people because I couldn't get my antennas to work or anything. But next year, I'll have a lot more freedom as a postdoc. And also, if you do have hummingbird feeders and you want to know what to do with the bats coming to them, like, do you leave them out for the bats? Do you take them down? I suggest tapering off the sugar water this time of year so that the bats can still get a drink, because they need a drink. They're hungry. There's nothing else for them to eat, but they don't depend on it and stick around just because of it. Like, we want them to move back south. We want them to be healthier. And if you do maintain a hummingbird feeder and you think you have bats visiting it, I highly suggest, since the spread of white nose is happening, to use alcohol or some like a high alcohol content over, I think, like 91% or over 70% alcohol to clean it, to disinfect it for white nose syndrome and potentially other viral things that might be transferred. That's another thing I'm going to try to study during my postdoc. Okay, questions? Or thank you, everybody. I'm not close my talk yet. And I'm on the chat too, so if people online have questions, I'll be monitoring that.
They catch the bats.
Oh, okay. We use I went over that slide really fast. We use mist nets. They're like badminton nets. Okay, yeah. Yeah. And for those of you who don't know what mist nets are, they're like a badminton net that go from the ground up. They're about 2 meters tall. We set them up in front of foraging sites, like in front of a hummingbird feeder or in front of roost entrances, and the bats can't see it at night and they hit it, get tangled, and then it gives us a chance to safely extract them and take measurements.
We've noticed quite a variation in sizes. Are we seeing young ones?
And sizes of bats?
Yeah, and they're all the long-nosed bats.
So they're all nectar feeding bats that
Yeah.
you can see?
Yeah, they come to our hummingbird field this time of year every night. They clean it out.
So,
But some of them are quite large, and we saw one the other night that looked like, the small brown bat.
so it's potential that insectivorous bats are just foraging on insects that might be feeding near your hummingbird feeder. When you see other like
No,
small,
this one's actually feeding in it.
okay, and then there are, they are like juveniles or first years.
And sort of.
So for the lesser long-nosed bats, their juveniles are brown and pelage, like their fur is brown, and they're a bit smaller, like their shoulders aren't as muscular yet, they're still building those muscles. So they are a bit smaller, where the adults are like gold, like they have this like goldish hue. So we call one dull and like one a bit golden. Yeah, and they are like very beefy. And I have at Russ's house, and I think Russ and Karen's place, and at Beth Lake and Ed Lake's house, I think I've caught reproductive males, and those are even larger, and they'll have a reproductive patch on the back, which you could see with the UV light. So I don't know if you have like a UV light on your feeder, and if you see it glowing, you're like, oh man, that's a reproductive male. And we have no idea why they're here. So I'm excited to figure that one out one day.
It's sticking around because it's warmer, or is there any kind of temperature mediated response for them going back down out?
So we used to suspect that it was like, well, the major suspicion is that it's agave triggers them leaving. And then they don't really like below 50 degrees Fahrenheit. They don't do so hot, but the roost is really where that 50 degrees matters, because when they're flying, they're staying warm. But I've gotten them here when it's like 40 degrees. I'm like, I don't know what's going on. Part of my like modeling that I quickly went over, the population modeling, is to try to figure out what that trigger is, like what's making them leave. But I think they're sticking around because they change their migration patterns because of anthropogenic food sources. And some of them are getting stuck here, and they're missing their window to leave because their nectar corridor is disappearing. And that might be because of climate change. I don't know. Maybe they weren't able to get enough protein to feel confident to make the flight back.
So should there be a different mix that one puts into a bat feeder than just nectar with sugar water?
I'm not sure yet. It's a question I've definitely asked people, but I'm not a dietitian, so I'm not great at that one. I've thought about it. That was like my first thought when the nectar corridor was disappearing. When I first came on this project, I'm like, oh, why don't we do this? But now that I'm thinking more from a disease ecology aspect, I don't think having an anthropogenic food source is a good thing, because these flowers with the agave, they wilt in 10 days. And there's thousands or millions on the landscape at a time. The chances of multiple bats visiting the same flower, unless they're foraging together, is very low. So then the chance of spreading disease is lower. So now if we have this anthropogenic food source that the bats like and are choosing over the flowers, then we might be just encouraging the spread of disease and potential zoonotic disease, since we handle these feeders as well.
But if they have a communal roost, wouldn't that be the same?
Saliva is different potentially. It matters how the disease is spread. Some things are airborne, some things are like if you drink out of the same cup as somebody else.
Speaking of roosts, do they do like the Mexican pre-tale where they have a maternal roost and a bachelor roost?
Yeah, so that map that I had earlier, a quick get to it, has different diagrams for different types of roost types. I'm just so proud of it. So the foraging sites are the stars, but then we have maternity roost, maternity and mating roost, transition roost, bachelor roost, and then that's the pet tag reader. So they have all different types of roost sites. Sometimes the roosts are both like in temporally, so like at different times of the year, it's a different type of roost because they're this migratory population. So like all the mating roosts are also maternity roosts at different times of year.
So that big hatchet roost is
It's a transition roost, where the maternity roosts are like Oregon Pike Cactus National Monument, Pinacante, just right across the Mexican side. I really want to go there. Mariana, Carmen, and Navachiste, and these Baja roosts are also maternity roosts. They're kind of all spread around. And there's more than on this map. This is just the ones that are participating in this pit tag reader study that we have.
Do you do any age distribution studies? Is that part of the study or not?
So we have first years and adults. And that's as much as we can age them without being extremely invasive and spending a lot of money. So usually they're at the maternity roost is where we have those age distributions, like first years. But then once they become adults, they're just adults and you can't really tell them apart. It would be cool to do that in the future, but bats research is very underfunded. So we gotta do what we can with the money we have.
You had that, I think it was the gold bat that was on the ground foraging.
Yeah, the pallid bat.
Yeah, I was under the impression that bats couldn't take off from the ground. How did
So
I manage
certain
that?
species can't take off from the ground, they'll have to climb up and then fall. But a lot of bat species have strong enough wings that they can hop. So
Oh.
actually do like a little hop and then they like start flapping and they can fly.
So what is it that you do, if you are to come to someone's house? Because I have bats.
That's a great question. So bats are leaving right now, so I'm not trying to take on any more sites and I'm leaving next week to go back to Colorado for 6 months. So but what I typically do is I'll set up a trail camera at your house, pointing away from everything so you can have privacy. I try to make it as private as possible. But I'll put it on if you currently maintain a hummingbird feeder, because I don't want to encourage people to put out hummingbird feeders too. I should say that first. You're already maintaining one. I can put a camera on it. I check the bat population. I try to come by once a month to check on the camera. And I can like kind of get arrival dates, activity levels at your house, and then departure dates. So right now I'm gonna try to go to all the sites. I haven't called you, but I was gonna ask if I could come by, check the cameras one more time. And I replace like the cameras, the batteries. I go off and I come back in the winter to see when did they leave. And last year, I think departure is November 16th here. So those numbers are really important for me to get for like when do they stop using residential sites. So I just try to get a good spread around town because sometimes I'll have bats at Russ and Karen's place. I won't have any at bats. So like they kind of are weird with how they forage. And the closest roost site that we know of right now, we just found it in 2021 was in the Burrows. So it's the newest. Oh, that's where you are.
And I've had bats this year and last year.
You would have the most consistent feeder site to track.
I have one bird feeder, one hummingbird. feeder.
Yeah, that would be great because I don't know how much information they're collecting at the roost at the Burrows yet, because that's another research group that's doing that one. So yeah, I'd be interested. I'll put my contact me slide back up. And I have a card if you wanted to grab that from me afterwards, but this or my new email is Mallory Davies at arizona.edu. Okay.
We have some nice refreshments out in the hall, and I would encourage you all to participate with that and stay in touch with Mallory with any further comments.
Go back.
Yes, I can make it, I think.